In this video, we will see how DNA replication is terminated in prokaryotes. I hope you've seen the replication initiation and elongation videos because I will refer to some of the content in those videos. In this one specifically, we will learn about termination sites and the Tus protein and understand how Tus and Ter stop the moving replisomes. Specifically, we will see how the structural complex of Ter and Tus protein achieves this task. Once we cover that, we will look at the actual mechanism of termination. Finally, I will give you an insight into the current debate on the termination system, where researchers are starting to wonder if the Ter and Tus system is really meant for termination of replication or if there is an alternative function for it. Let's start with a couple of basic things to bring everyone on the same page. We're going to discuss the E. coli bacterial chromosome, and bacterial chromosomes are mostly circular with some exceptions. And this Ter-Tus termination system is generalizable across all gram-negative bacteria. This means that not all bacteria have this system, but have evolved similar mechanisms. Alright, since the chromosome in question is circular, let me sketch out the generic picture of the replicating bacterial chromosome. We have already talked about this in the trombone model of elongation, where two replisomes are recruited and they start moving in the opposite direction as they replicate the circular chromosome. We will call these bad boys replication fork 2 and replication fork 1. And the point of start and recruitment of the replisome is the origin of replication. Following initiation, they will elongate, and we have discussed how that happens. Now, as they move in opposing directions, they will eventually converge at some point where replication has to be terminated. And that point is called the replication termination zone. This zone is divided into two parts, where one part has 5 termination sites facing the same direction, and the second part has another set of 5 termination sites facing the same direction but all opposing the other side. This entire replication termination zone is around 350 kilobases in size. Some textbooks also refer to this as the terminus replication in case you come across some different name. So the entire zone has about 10 sites, which all look roughly the same. They're not palindromes, so that means they have some sort of directionality to them. And all these 10 sites are give or take somewhere around 23 bases in length. So each of these arrowheads are 23 bases in length. And each of these sites is called a termination or Ter site. The key thing to remember is that these are directional. By the way, not super important to remember, but these sites have names, like J, G, F, B, and C, which all face the same way, and then you have A, D, E, I, and H, which all face the same way. So conventionally, you call them Ter-J, Ter-G, or so on until Ter-H. Just hang on to this directional part for a bit, and we will come back to it in a short moment. First, let's talk about the Tus protein. The Tus protein is the product of the Tus gene, which is about 309 amino acids in length. Tus stands for Terminator Utilization Substance, and its function is to bind the Ter sites. Specifically, one Tus protein, so as a monomer, it binds one Ter site. And we have noted that each site is around 23 bases in length. But the Tus protein only binds 15 bases out of that 23 bases. This points to the fact that it has a sequence specificity. The complex of Tus and Ter function primarily is to block the moving replisome. And those are the two forks that we mentioned. More specifically, the Ter-Tus complex destabilizes the moving helicase, which destabilizes the entire replisome. Essentially, the Tus protein makes the Ter sites directional. So the directionality of this entire replication termination zone is dictated by the Tus protein binding. Let's understand this concept of directionality via an example, and I will take Ter-B as the explanation. Ter-B is about 22 bases in length, and here's the complete sequence of the Ter-B. Now onto this DNA site, one Tus protein will bind, and it will bind only at about 15 bases out of these 22 bases. The core of Tus protein has negatively charged beta sheets that deform the typical bDNA conformation. And this allows access into the minor and the major grooves. And this is where the sequence-specific binding occurs. One specific sequence contact is made at the 6th position cytosine in this particular region. This cytosine is then flipped out into a binding pocket of the Tus protein, and arginine is responsible for directing that interaction. This particular conformation between Ter and Tus is called a strong locked conformation. That only happens due to this cytosine at the 6th position, and this provides the Ter sites its polarity. By the way, this flipping of the base is similar to RNA polymerase binding to the Tara box. Notice how I made these sites into arrowheads? So this particular site can be overlaid with an arrowhead, and it looks something like this. But what does it mean? The polarity or directionality is understood in the context of a moving replication fork. In the context of Ter-B, the replication fork 2 is traveling in this direction, and the fork 2 can destabilize this Ter and Tus complex, and it will pass through Ter-B site, which means that this side of the site is permissive to the replication fork movement. The opposite side has the replication fork 1 coming in the direction. In this context, replication fork 1 cannot destabilize the Ter and Tus complex. So it cannot pass the termination site this way. The complex prevents the replisome movement from one side only, and we call this side to be the non-permissive side. This is just one example, but let's put all this information together and understand how replication termination zone is established. All these 10 sites are bound by the Tus proteins, and the replication fork 2 is moving along, and it will destabilize and pass through all these termination sites because of the non-permissive polarity. But as it gets to the other side of the termination zone, it will hit the permissive side of A, D, I, and H. So it can be stopped by any of these sites. In contrast, you have replication fork 1, which is coming from the opposite direction, and it passes through A, D, I, and H termination sites because of the non-permissive polarity, but it can be stopped by any of the sites from the other end. So that is the important takeaway concept in the directionality of the termination zone. Now that we have discussed how Ter and Tus complex forms and how it functions, let's get into the termination mechanism. Let me quickly fix this chromosome first. Now, the key fact to put forth is that the middle of the termination zone happens to be in between C and A site. And this is also roughly exactly the opposite of the origin of replication. So both these points are roughly equidistant. Before we get into the mechanism, let's see what are the possible scenarios in replication termination. And I will primarily focus on the termination sites C and termination site A. Now let's get into the first scenario. In this scenario, the replication fork 1 arrives at the replication termination middle point by crossing the Ter-A, and it hits the permissive side of Ter-C. Now it can either wait there or be released. And let's just formalize the scenario. The fork 1 reaches Ter-C first, and either it waits for fork 2 there or disengages. In our second scenario, the situation is reversed, meaning that fork 2 reaches Ter-A first, and either it waits for fork 1 or it disengages. In the final scenario, we take into the consideration that the middle point is between termination site C and A, and that the speed of both of these forks is the same. So if they start from the origin at the same time, they will meet in the middle if they're moving at the same speeds. To formalize this, we can say that the fork 1 and fork 2 meet at the middle of the termination zone. In the previous two scenarios, one of the forks is moving faster, so they don't meet. But in this third scenario, we assume equal speeds. Now, there are two outcomes. Either the replication forks collide into one another and disassemble, or they can cross and pass over each other. And apparently this passover outcome seems to be the most common outcome. And that means it is the most common way to terminate DNA replication. Now the question is that if they pass over each other, then how does the replication terminate? Now it is time to expand on this scenario and understand how passover can lead to replication termination. I will again expand on this same position, but now we will get into the nitty-gritty details of the replosome as it moves on the Ter-C and Ter-A region. In this one, please note that I'm not going to point out the leading and lagging strands. I hope that you have understood the trombone model of elongation for that content. Alright, so we have got the permissive P and non-permissive NP sides of each termination site. As we have said, when these two replication forks move, they will destabilize the Ter and the Tus complexes because of the permissive polarity, and they meet and pass over each other in the middle. Now, when the replication fork 2 passes the termination site, the fork 2 will have synthesized or copied the Ter-C site. And now fork 2 is sitting in the middle. Notice that it is not attached to the bottom strand. It is only connected to the top strand. And it is no longer in the replication fork form. That is the key difference when the two forks meet. So that's the movement of fork 2 over the Ter-C site. Similarly, fork 1 will have moved along the Ter-A site and copied the Ter-A region. Now, in front of fork 2 is the newly copied DNA with the Ter-A, which is also occupied by the Tus protein. The same story occurs on the bottom strand. As the replication fork 1 moves along and passes over the fork, it will have synthesized or copied the Ter-A behind it. And on the other side, as the fork 2 moved along, it copied the Ter-C region, which is now again occupied by the Tus protein. The replosomes are not in their traditional replication fork form, so they will not make any new DNA at this point, but instead they just keep moving in their respective directions. But both will stop as they hit the non-permissive side of the Ter-Tus complex. But notice that there is this double-stranded DNA between the replosome and the termination site. Since the replosome at this point is simply a helicase as it moves, the helicase unwinds this double-stranded DNA, but only until it reaches the Tus-Ter complex. And when it hits the non-permissive end of the complex, the helicase is released. Finally, you're left with this overhang single-stranded DNA, which is called the flap. And this small portion of non-copied single-stranded template as a gap after the replisomes crossed each other, because as I said, they are not in a fork form after crossing. The replosome is pretty much like a moving helicase. Okay, that seems like a lot of information, but let me simplify this. So after the release of helicase, you're left with some 3' end flaps and empty gaps. Now, this is exactly the same type of thing we saw in our elongation video. To solve this problem, DNA polymerase 1, which has the Fen activity, will clip off these flaps and fill the gap left by the helicase. And finally, the ends are stitched together by the ligase. This is the same mechanism we noted in the Okazaki fragment maturation when we discussed strand displacement. You should check the trombone model video for more details on how this works. And that is how the replication termination in prokaryotes is thought to work. But I said in the beginning that this process is only true across gram-negative bacteria, which means that the Ter-Tus system of termination is not universal. Sure, there are analogous systems in other bacteria, but the point is that the bacteria is fine if you get rid of this termination system. And this has brought the termination system into question, and specifically researchers have been asking whether Tus and Ter is really functioning as a termination system, and if termination function is a byproduct of some other function of Ter and Tus complex. And as I said, we can delete the Tus protein, which makes the termination zone meaningless. And to our surprise, if you do that, the bacteria is unaffected by this loss. However, there are issues with chromosome segregation. And this has led to a hypothesis that the Ter-Tus system is probably a system to prevent abnormal DNA replication initiation, which results in segregation issues. To understand this, we have to understand what would happen to termination zone after passover if Tus protein is not present at all. For explanation and simplicity, I will take this bottom strand as the example, but note that the same mechanism will happen on the other strand too. Now, as the helicase moves on the template, we will observe that the flap is getting longer, which simply means that the double-stranded DNA is getting unbound. This is because there is no Tus protein to establish the termination zone, which means that the non-permissive and the permissive idea cannot be applied here. The termination sites are totally useless without the Tus protein. So in this example, the helicase 1 has moved past Ter-C and is going to cross the Ter-B, and this flap will keep getting longer. But at some point, it will be long enough that the helicase can use the single-stranded flap and the bottom strand as templates and restart DNA replication. So the flap is then converted from a single-stranded template to a double-stranded DNA. And now it is a template for leading strand synthesis. Since bacterial chromosomes are circular, this end of the flap is not closed in a loop or a circle. So bacteria thinks that this is a DNA breakpoint and tries to fix it. So the flap will strand invade and a process called homologous recombination will start. I will not go into the details, but proteins like Rec A, B, C, and D are involved in initiating this abnormal repair. And here is a quick sketch of what this strand invasion will look like. Essentially, the recombination leads to the resolution of holiday junctions. Again, you don't have to know this, but if you have studied meiosis or cell cycle, it is exactly the same thing. And after resolution, the strands are exchanged and it will look something like this. Now, the flap does not exist and the DNA is in a closed loop. But it looks like there is unbinding that has occurred. And this loop is recognized by primosome proteins, which think that this is a replication bubble, and they help recruit a helicase into this loop. And this leads to establishing a second replication fork in this position. Again, this is an example of the bottom strand. The same thing will happen on the other strand on the Ter-A side. So, in the absence of Tus protein, we have converted the termination zone into a replication initiation zone, which typically is the origin of replication. So, the supposed termination zone has become abnormal. So functionally, the real role of Tus-Ter may be to prevent this sort of abnormality, and termination system is just how it is observed, but not how it evolved.